Good morning. My name is Elaine Bearer, and I'm making this video in Pasadena, California, while I've been working on my projects at California Institute of Technology. Today, I'm going to talk about axonal transport and my personal quest for the biological basis of cognition and emotion. I'm going to try to share my screen now. So, axonal transport in the search of biological basis for emotion. My name is Elaine Bearer. This is for the ICBN webinar scheduled for July 24th in London, from London. This is a picture of New Mexico, and my professional appointment is a Harvey Family Endowed Professor with tenure in the Department of Pathology at the University of New Mexico School of Medicine. Some of this work was also done at Caltech, where I am right now, and at Keck School of Medicine and University of Southern California. Recently, the news has come out with a very exciting report of adversarial collaboration for testing hypotheses. This was reported by Christoph Koch and several other people at a big meeting at NYU in New York about a month ago. Christoph Koch published the idea for this adversarial collaboration in Science in 2021. They were interested in testing two opposing hypotheses of how consciousness works. Consciousness obviously is the first step for cognition, but consciousness may not be the first step for emotion. Emotion may be pre-conscious. So we're looking at two different types of mental processing if we search for biological basis for both emotion and cognition. What their two hypotheses had a global network theory, workspace theory, where activity would be distributed, or a temporal dynamics of a single integrated network, or from the integrated information theory. The results of this adversarial testing were showing that both of these theories have merit, and they may occur simultaneously or for different problems. So they used MEG, EEG, fMRI, and electrophysiology in epileptics to measure activity. They measured activity throughout the brain so this is brain-wide work. They needed to see the whole brain, or they couldn't address these theories. It was all done in human, so that the types of manipulations were limited. They found that deeper brain structures shown in this figure from their recent Cogitate Consortium preprint in bioRxiv that was just posted a couple of weeks ago. They showed that each modality coincided. They got the same answer with each of these modalities of testing the brains. They tasked the humans with video games and object viewing, and they looked at the results. What they found was that both theories had merit. There will be ongoing studies, and obviously one of the things they said was, while applauding the present efforts, they're eagerly awaiting results of other adversarial testing in animal models where causation can be addressed. So I'm now going to turn to what the definitions of cognition are. So we have actually two schools of thought. Again, we have cognitive neuroscience, which is the mental process of knowing and thinking, and we have cognitive impairments that neurologists use to test people for cognitive impairment, as in dementia, as in neurodegenerative diseases, and other forms of cognitive failures. So we have two definitions. We don't yet have an adversarial collaboration, however. For the second definition, we have a lot of biological brain data. This comes from our Alzheimer's centers. There are 34 Alzheimer's centers distributed across the US, funded by National Institutes for Aging. Alzheimer's is a neurodegenerative disease. On the left, this is a human brain. On the left is a normal brain. And on the right is a person who was diagnosed with Alzheimer's disease, which means they had both dementia and they had the classic pathological signatures in the brain as viewed histologically. Those signatures are plaques and tangles. This is what plaques look like in hematoxin and eosin. And this is what A beta looks like when it's deposited in these plaques. We can also stain for the tangles with antibodies to phosphorylated tau protein. And this shows us tangles, which are cages of aggregated tau in filamentous form that cage the neuronal nucleus. So these are Alzheimer's brains, and these are the pathological signatures of those brains. A lot of work has gone into identifying those proteins. The latest treatment is targeting the A beta and its deposition. It now turns out that A beta deposition may not be correlated with dementia. In fact, many people may have A beta depositions and show no cognitive impairment. So how might these pathological features impact cognition? Well, they could affect circuitry by altering rates of axonal transport. They could affect networks and connectomes, and they could affect the hidden brain state, latent or hidden brain state. My contention is that they do all three of these things, and that each one has a slightly different clinical feature. I've spent probably the first middle third of my research life studying axonal transport. Axonal transport is the process by which those molecules synthesized in the cell body are transported out to the synapse in the long neuronal axon, or back from the synapse to the neuronal nucleus. The retrograde transport informs the nucleus to survive or sprout, and the anterograde transport mostly provides the material for maintaining the synapse and for transsynaptic signaling. The axon of the North Atlantic long-fin squid, Loligopelii, extends from just behind the eye all along the mantle. It's a very large axon. It measures 1 millimeter in diameter, unlike the axons in the human brain, which can be as small as 500 nanometers. The axonal transport along the neuronal process depends on the cytoskeleton, the microtubules, and actin filaments. Tau protein is a microtubule-associated protein, and when it is abnormally phosphorylated, it disrupts this process. If we look into the axon, we can see how this axonal process works. This is a video I made looking into the squid giant axon when we were using it as the basis for our study of the molecular mechanisms of axonal transport. I did most of this work with Tom Rees, who is a director of a large lab at National Institutes for Neurological Disease and Stroke, and was the senior author of the first paper to discover the kinesin 1 protein, which is the molecular motor that drives this transport in neurons. Sorry, advancing is not easy. So we decided, after studying the squid axon and understanding the molecular mechanism of that process, to see if we could image axon in a living brain. I chose to use magnetic resonance imaging for this, and manganese, because manganese is a paramagnetic ion that enters these transport vesicles, and we can follow transport from various localized injection into the brain into other regions throughout the brain by doing whole brain imaging in an 11.7 Tesla Brooker MR scanner. This diagrams the essential approach we used. Localized track tracing allows manganese to be taken up by active neurons in a region of the brain, and then we can watch where the manganese travels to by its T1 weighted MR hyperintensity. Our methods include precise localized intracerebral injection, time-lapse MR imaging, alignment of the images, and 2 levels of computational analysis, one which is data-driven and the other based on region of interest of candidate regions. So we take, in some of our series, we have taken 4 MR images. We take a pre-injection image, so there's no manganese, no contrast. Then we inject the manganese in this precise location, and we take multiple images across 24 hours. We sacrifice and then look at a gold standard type transport marker, rhodamine, dextranamine, which we can see after sacrifice by microscopy. So this is what the experiment looks like. We injected in CA3 of the hippocampus, and we followed the manganese across, progressing to the medial forebrain, which is thought to be the place where memory is consolidated from CA3. We looked at this in 2 different genotypes, a wild type genotype, and then its littermate, in which mimics our human Down syndrome, which is a trisomic, and has 1.5-fold excess amyloid precursor protein, the precursor for the A beta plaques. We found, we had already described in SQUID that amyloid precursor protein was part of the machinery that carries vesicular organelles from the neuronal cell body to the synapse. And so we wanted to see if you have too much of that, what that might do to the delivery from the neuronal cell body to the synapse. And essentially what this paper showed, with a lot of computational analysis, that when you have 1.5-fold excess APP, you get more robust transport, as in the blue image overlay here, compared to the wild type, which is in red. So excess APP increases and improves transport. This is wild type APP. This was the normal APP, not the mutated forms that produce Alzheimer's in human. We then found that APP is necessary for normal rates of axonal transport. We published this in 2011. So when you knock out APP, now the red is the knockout, not the excess. And the knockout, you can see here very dramatically less transport, and we could measure that and statistically compare the intensities. And you can see in these 2 important regions where transport is required for the manganese to arrive, you get much less, where in wild type you get 23% increase in transport. In the APP knockout, you only have 1.9. That's the contralateral hippocampus. And in the septal region that I showed you for the Down syndrome, you might get a 45% increase in signal in wild type animals, and 0% in this particular region of interest in the knockout. So APP, excess APP increases transport, absence of APP decreases transport. This is very interesting as it relates to cognitive performance. So we decided to create an Alzheimer's-like mouse. This mouse expresses an abnormal human APP, and at 6 months, we see that it has made plaques, and at 1 year, it's made lots more plaques. And we can see that a wild-type littermate has no plaques. So so then we measured transport in these animals, and we wanted to look at whether aging has an effect on transport. And we found that even in a wild type mouse, as is shown in green here, and an old wild type mouse shown in blue, there's definitely decreased transport in this hippocampal to forebrain circuit. If we add these plaques to the situation, we get even more decreased transport, so that there's hardly any transport in the old APP SWIN. What's very interesting is that this mouse is still alive, moving around in the cage. They don't die any sooner than the other mice. They just have this transport defect. And here we have a three-dimensional rendering of these two situations, the old wild type and the old APP Swedish Indiana mutated animal. And you can see the transport from the yellow hippocampus goes to the medial forebrain, in blue from the old mouse, and in red from the one with plaques. It's much decreased. These are statistical analyses. You can read the whole method, which I'm not going into, but essentially we have 12 mice in each of these 4 cohorts, and this is a statistical parametric mapping of the intensity changes from the manganese. So transport from CA3 in the forebrain is diminished in the older mouse and further diminished by the presence of plaque. Well, then we wanted to see, was it the plaque alone or was it the overexpressed mutated protein? So we created an interesting mouse in which the CAM kinase promoter drives the TET-off gene, which then activates the expression of the mutated Swedish Indiana APP through its TET-off promoter. This interaction can be blocked by doxycycline. So we could treat the mice with or without doxycycline and turn on or off this gene, this mutated APP gene. And what we found was with no doxycycline at 6 months, we get the scattering of plaque. If we turn off the expression of APP, the plaques persist, they don't go away. Joanna Jankowski has also shown that using a similar mouse. When we raise the mouse in the presence of doxycycline, there's no plaque, and we can detect that with a blot for A beta, and we have no A beta, or no detectable A beta. And if we take a wild type mouse that doesn't have this gene, it has no plaques and no expression. And then we can look at transport under these different conditions. And we color code that with plaque and overexpression of the mutated protein in yellow, plaque alone and red, no plaque, but excess APP in blue, and the wild type. And as we expect, even though it's mutated APP, we see a lot more transport into the medial forebrain in the animals with overexpressed APP, and we see diminished transport in the animals with plaque. So in the situation in humans and in the mouse with both, these two excess transport and decreased transport overlap, and we get kind of a normal picture because they mutually correct the deficits. So the conclusion from this is that APP and kinesin 1 are each required for normal transport rates. Excess APP drives higher rates of more distal activity. Transport rates decrease with aging. Amyloid plaques with neurofibrillary tangles, that's phospho-tau, further decrease rates in young mouse. I didn't show you that, but it's in the publications that I have cited. And human mutations in APP produce products that interfere with transport independent of plaques. So, therefore, transport circuitry defects could contribute to cognitive impairment. We've adopted another study, and this is to look at forebrain projections. This is a picture of Phineas Gage, who famously put this iron bar through his forebrain and lived to tell about it. He lost executive function, but could do many other things. He lived for 5 years or more after this accident. This part of the brain, therefore the forebrain, has become a focus of much interest. This image is from a paper by Koob and Falco, who are both NIH directors in the US. Koob is director of the Alcohol Abuse Institute, and Nora Falco is director of National Institutes for Drug Abuse. Both of those institutes are focused on what a drug does to this executive choice pathway. Rodents have been a model of choice, and the rodent brain has been mapped to interesting similar regions in the human prefrontal cortex. I think this will work. So we have entered into a study of the prefrontal cortex, again, using our manganese, injecting into the prefrontal region of the mouse brain. And these injections are in the infralimbic cortex. We can localize that. We've looked at 72 mice, 12 in each of these data sets, a wild type and a cert knockout, a NET and its cert knock and its wild type littermate, and a DAT knockout and its wild type littermate. The hypothesis is that these monoaminergic systems regulate the activity of the prefrontal cortex. We created a segmented atlas with over 100 different segmented regions. And then we could map where the transport goes. This is not published yet. It's in process. Hopefully it will be submitted in the next month. And what we found is when we inject in the forebrain in this localized region, we get strong signal in the striatum in all of these regions at about 8 hours after injection, which is about the amount of time we have measured manganese can travel along axons to distal destinations. We measured that in the visual system. We now want to know why this is going to all those regions, and we think it's because the activity in those regions has been changed. So this shows a diagram of what we're doing here. Instead of localized injection, we're giving an intraperitoneal injection. When a neuron is active, it takes up the manganese and that uptake throughout the brain in various locations can give us a manganese signal for where the neurons are active throughout the brain-wide. So we take a pre-manganese injection image, we take an image 24 hours after injection, and then we repeat at 9 days. We use a complicated computational pipeline to process these images so that we can extract statistically significant information. We've published this, so I won't go through all the steps, but suffice it to say, we have to remove the brain because these are all living mice that we're imaging. So we strip the brain in an automated process that we designed, published in 2017. We then align the brain using a warping that uses the minimal deformation atlas or a template and applied to the brain so that we're not warping based on the, sorry. We're not basing our warping on the hyperintensity manganese signal, but on the actual anatomy of the brain. And then we get data stacks of 12 for each animal in our post-warped, processed, and ready to analyze image stack. For processing, we have been using statistical parametric mapping by comparing post-manganese images with their pre-injection in paired t-test. So the paired t-test gives us quite a lot of strong statistics. We usually use 12 mice per data set, and we've been using TMT, a predator odor, to alter the brain and see what the alteration in brain neural dynamics are. This was published in 2020 in Neuroimage. And now we're working on a new paper looking at how early life adversity affects the brain state. So we've come to the conclusion that what we're measuring when we look at the whole brain with this MR manganese enhanced imaging is a distributed pattern of brain activity that actually changes under specific conditions, such as predator odor or the recovery afterwards, or no predator odor when the mouse is normally moving in its home cage. Recently, and just last month, May 23rd, no, I guess 2 months ago now, Green et al. Published a very interesting paper in Trends in Neuroscience where they defined what a brain state is. They said it was the product of a specified cognitive or physiological state. It's characterized by a widely distributed pattern of activity, which we can see with our MR imaging. It affects the future physiology and or behavior of the organism, and it likely results from neuronal activity that propagates across the structural scaffold of the brain and is modified by neuromodulatory tone. So this gives us this idea of brain states. one network, but many outcomes. So I diagrammed this very manually, very similarly. Let's say you have a diagram, a network that includes the BSD, PFC, amygdala, hippocampus, but the BSD is very, very busy and the rest are not busy. This would give you one outcome. You could change the amount of busyness or activity in these different nodes within the network, and that would change the output, the feeling that network, the thought that network, the music, the poetry that network gives as an internal brain state sensation. These networks could be balanced by neuromodulatory systems like the dorsal raphe, which sends serotonin throughout the brain, the locus coeruleus, which sends noradrenergic signals, or the ventral tegmental area, which sends dopamine. So we're trying to figure out whether this is true, and we've been using our powerful computational image analysis to align huge data sets, over 100 images of animals that have received injection in the forebrain, and how does that tracking, and these are two different locations for the injection, how does that track into the brain? We find that the ACA is greater than the infralimbic in the locations where we show blue, and the infralimbic region is greater than the ACA in the regions we show red. So now what was the brain activity and how does that correlate to these projections? And I'm just showing one here. This is the correlation between the ACA projections and brain-wide activity. And we see that in the nucleus accumbens, the projections from the ACA, which are in yellow, and the brain-wide activity correlate, but not many other places. Here in the hindbrain, yes, but not here in the hypothalamus. So it will be of interest to further pursue how these localized neural activity dynamics and brain states correlate with projections. This is how Alzheimer's affects the brain. These are the plaques and tangles here stained with silver. Alzheimer progresses from the entorhinal cortex into the hippocampus, which is what I've been somewhat showing you, and then up from the hippocampus into the cortex. As these stages evolve, the phospho-tau has progressed up into the entire cortex. This is published by Brackett in 1995. Tal et al. Showed that the similar progress, but in an opposite direction, occurs with A-beta, where it starts in the cortex and progresses into the limbic system and deeper structures. So this is my team, and I'm going to wrap it up now. I hope people will send me emails. I put my email on the slide, so you can go back and scroll back on this video and find my email address, and let me know your questions. Thank you very much for your attention.